It's a real pleasure to be here today. Can you hear me in the back? Is this working? Yes? Okay. What I'd like to do today is to give you some background, although you heard a lot of it from Louisa. I will give a general background of where we became interested in telomeres and telomerase. And then I'd like to move into some of the implications that we've been following up of the roles of telomeres and telomerase in disease. So to start at the end, that is the end of the chromosome, what we're very interested in, if you look inside the cell, you have the chromosomes, and probably most of you in the room study the various components that are along the length of the chromosome. But what we're going to focus on here is the telomere, or the part that protects the chromosome end. So telomeres have two essential functions. First, to protect the chromosome end, to protect it from degradation by nucleases, to protect it from recombination and joining. Telomeres also have to maintain the length and the integrity of the chromosome, and we'll be talking a lot about this length maintenance. So I didn't put this slide in here because I knew there was tetrahymena here. This is really the slide that I always use. So the telomeres were first identified in the ciliated protozoan tetrahymena thermophila, where Liz Blackburn and Joe Gall first determined the sequences that are present on the ends of chromosomes. And what they found is that there are very simple, tandemly repeated DNA repeats. And as it's turned out, this is conserved throughout evolution. All eukaryotes that maintain their chromosomes, the linear chromosomes, have a variant of the particular simple, tandemly repeated DNA sequence at the end of the chromosome. And this is important to use tetrahymena because if you want to understand how chromosomes are maintained or what their structure are, you go to the source of the organism that really knows how to do that the best. And because tetrahymena has so many chromosomes, it was the ideal source to be able to understand this problem. So telomeres are made up of these simple DNA repeats. So what's shown here is just the double strand region of the telomere, and there's a small region at the end of the chromosome, which is single-stranded. And I'll be talking a lot today about the vertebrate telomere repeat, and that sequence is TT-AGGG. This is just one nucleotide different from the tetrahymena, which is TT and then 4 Gs. So in order for a telomere to function, you have to have both the DNA sequence as well as the proteins that are bound along the length of the DNA sequence. And in the vertebrates, that is termed the Shortening complex. Different organisms have different terminologies for this. But basically, there are proteins that bind to the double strand of the DNA, and proteins that bind to those proteins, and also protein that specifically binds to the single strand region that's at the ends of the chromosome. And together, the DNA and the protein have to provide these functions of protecting the chromosome end and maintaining the length of the chromosome. So it's been known for a long time since we've understood how DNA replication normally works inside of cells, that the replication machinery can't proceed to the very end of the chromosome. And so as a consequence, if this is most of the chromosomal DNA, and then this is representing all of the telomeric DNA repeats, as a consequence, every time the cell divides, there are a few repeats that are lost from the end of the chromosome. And what I'm going to show you is that these repeats basically provide a buffer zone. That is, they don't code for anything. You can lose most of these repeats without any consequence. And it's not until you lose this buffer zone that you have any consequence to this shortening. But of course, this can't go on forever because your chromosomes would be continuously shortening and shortening. And so we were very curious about how the chromosomes can be maintained. And that is when Liz Blackburn and I were able to identify this enzyme that we called telomerase. And what this does is if during replications there's some loss of the telomeric repeat, the telomerase enzyme can then add sequences back onto the ends of the chromosome. So what happens is that there's shortening during DNA replication and then lengthening due to telomerase. So the telomerase doesn't fix the problem of the shortening, but rather it balances it out. So the telomeres are in a continuous state of some shortening and some lengthening and shortening and lengthening. And they are maintained around a specific equilibrium set point. So the way that telomerase does this is it's this holoenzyme, and what's shown here in this dark gray line is the RNA component of telomerase. And within the RNA component of telomerase, there's 1.5 repeats of the telomeric sequence that has to be added onto the ends of the chromosomes. So this gray here is what we call the TERT, or the telomerase reverse transcriptase. So this is the catalytic polymerase that adds repeats onto the ends of chromosomes using this RNA template. So the terminology here is TERT, telomerase reverse transcriptase, and we call this imaginatively the telomerase RNA, or TR. In various different eukaryotes, there are a set of proteins that will protect the 5' end. What's shown here is the mammalian complex, but different organisms have different kinds of 3' ends to protect and stabilize that RNA. So we were curious about what would happen in a cell if you didn't have telomerase activity. What would be the consequence of the continuous telomere shortening? And so what we were able to do is to take the mouse telomerase RNA and generate a mouse in which one of the 2 copies was eliminated, and take 2 such heterozygous mice and cross them together. And of course what we get is the wild type mice, the heterozygous mice, and the completely null animals. These animals were born in normal Mendelian ratios, and there was nothing apparently wrong with these telomerase null mice. And so we call these the G1, or the first generation in the absence of telomerase. And so since we knew that there was a buffer zone on the ends of the chromosomes for the telomeres, what we did was to cross these first generation telomerase null animals to each other, cross the G1s to the G1s, and then we generate the second generation telomerase null animals. Those mice were perfectly fine and viable, and so we did the next cross to the G3. You can see where this is going. We then did G4, G5, and G6. We don't go any further than the G6, and I'll show you the reason that we don't. These animals are infertile, but also have a number of other problems with cells that normally divide many times. But what I'll be talking to you about is the fact that these G1 animals have no consequence, and it's not until the telomeres get short in the G5 animals that is important. So of course what we wanted to do is to follow what's actually happening to the telomeres with each generation of breeding these telomerase null animals. So we used this assay, which is called QFISH, for quantitative fluorescence in situ hybridization. And what's shown here is a mouse metaphase spread. And you can see mouse chromosomes have their centromeres all the way at one end. And so the chromosomes look like these little boomerangs. The blue is the DAPI staining of the telomere of the chromosomal DNA, and the red is hybridization with a probe to the telomere repeat. And you can quantitate how long a telomere is by how much probe has hybridized. And we need to use this assay because in the mouse, where we're doing this experimental system, the restriction fragments are around 100 kilobases long. And it's very difficult to see a small change in size of a very long restriction fragment. So this assay of measuring the fluorescence intensity turns out to be much more specific to see changes in telomere length. So what we do is we quantitate the signal on each one of the chromosome ends. And when we do that, when we look at the parents here, what you see is the fluorescence intensity plotted across the bottom here, and the number of ends that have that intensity. So you see this nice frequency distribution, and this frequency distribution represents the normal shortening that occurs and then elongation by telomerase. So telomerase normally maintains this established equilibrium telomere length. But then with each generation of breeding, here's the G2, the G4, and the G6, you can see that there's significantly less signal on the G6. So with each generation of breeding, there's a progressive decrease in the telomere length. So when we now look at the phenotypes in the mice as we're breeding these generations down, what we find is at the cellular level that in the G4, G5, and G6, we see increasing either cellular senescence, that is, the cells just stop dividing, or apoptosis that occurs in these cells. And so what we know is that it must be the short telomeres that are causing this effect, not the absence of telomerase. Because it was the absence of telomerase that caused the apoptosis, you would see that in this first generation. So one of the important take-home lessons from this is that it's the short telomeres, not the absence of telomerase, that cause cell death. So what actually happens to the organism? At the organismal level, what we see, I mentioned that there's infertility in the late generations. So if you look in the testes at the seminiferous tubules in the wild type, you see these normal tubules where you have the meiosis around the periphery and then the spermatids moving in. And in the G6, you see these empty tubules because there's apoptosis in the early meiotic precursors. We also see intestinal villus atrophy. So normally in the intestinal lining, you see these very nice villi with the stem cells that are here at the base of the villi. And in the telomerase knockout, about half of the small intestine is completely devoid of villi. And there's also bone marrow failure. So normally you see these cellularity within the bone marrow, and you can see this empty bone marrow. So these were cells of high turnover, and so it was something that might be expected due to the increased number of cell divisions that these tissues undergo. So how is it that a short telomere can cause cells to die? And I'm going to summarize what's been shown by a number of different laboratories in the telomere field. And that is that when telomeres get to be very short, they resemble damaged DNA. So what normally happens in a DNA damage response is that if you have the normal DNA and you cause a break somehow, that double-strand DNA break is recognized by a set of proteins that sets off then a cascade of signal transduction, which then goes through P53, and depending on the cell type and the amount of damage, the cells undergo either apoptosis or cellular senescence. And so what work from a number of laboratories has indicated is that when you have a normal telomere, what it does is it protects the chromosome end from being recognized as a double-strand break site. When that telomere now gets to be too short, it no longer has that protective function, and now the telomere will signal a DNA damage response. And that DNA damage response goes through ATM, and there's a signal transduction, requires P53 for the cells then to undergo either apoptosis or senescence, depending on the cell type. So we are very interested in understanding how this plays out in terms of disease. And what I'll tell you about is that there are two different aspects to the role of telomerase in disease. And that's because telomerase is required for all cells that have to divide many times. So there are going to be two stories I'll tell you here today. One is that during normal tissue renewal, you have tissue-specific stem cells that have to renew themselves and generate pluripotent precursors, and then all of the other cells that arise therefrom. Also, in cancer, you have specific cells that are undergoing the stem cells, which are self-renewing, and then the differentiated products. And if there's a change in one of those cells, which causes this now to become a cancer cell, that cell requires telomerase in order to be able to continue to divide many, many times. So both cancer and normal tissue regeneration require this maintenance of the telomere ends. So I'll tell you first a little short story about cancer, and then I'll focus on some more recent work on the role of telomeres in normal tissue-specific stem cells. So it's been known for a number of years now that telomerase is activated in cancer cells. So if you compare tumor tissue to a normal tissue, and about 85% of human cancers have higher levels of telomerase activity. The remainder of the 85% use an alternative method to elongate telomeres. So telomere maintenance has to occur in all of the cancers. And I don't have time to really get into the details of that today, but I'm happy to talk about it later on if people are interested. So we've known that human cancers have activated telomerase. So we wanted then to use our telomerase in all mice to ask a question about what would happen if the tumor cells do not have telomerase and can't be maintained. So David Felder, when he was a graduate student in the lab, set off to do this experiment where he crossed the telomerase null mice to emu-MYC mice. So these are mice in which the MYC oncogene is driven by the emu B cell specific promoter. So these mice have been well characterized, and they get B cell lymphoma with 100% penetrance due to the MYC oncogene. So what he did was to cross this MYC onto the telomerase heterozygous mice. and then cross those mice to get the G1 mice that also contain the MYC transgene. This, of course, generates the first generation telomerase knockout mouse that has the MYC transgene. And so he had to take those mice and cross them to other telomerase null G1 mice, and then generate the whole line of mice for 6 generations. So as a graduate student doing a 6th generation mouse experiment, he was very brave. But what David was able to find is that because of the telomere shortening with this increased number of generations, there was a fairly striking effect in the 6th generation mice. So what's shown here is the Kaplan-Meier survival curve. What's plotted across the bottom is the number of days, and this is the percent of mice in the colony that were alive. So at the start, if you follow the black line here, these are the mice that are transgenic for the emu MYC, but are wild type for telomerase. And what you see in this black line is that there's this survival curve, and at about 100 days, half of the mice have died, and they've died all from B-cell lymphoma. And eventually, 100% of the mice die. Now, if you look at the emu MYC, which are null for telomerase, but they're the first generation, what you find is that there's a very similar survival curve. So these mice have no telomerase, but they still have reasonably long telomeres. In contrast, the emu MYC mice that are null for telomerase in the 6th generation have a dramatic increase in their survival. And what's shown here is in each one of these little ticks is David went in and looked in these mice, and what he found was there were small microlymphomas. So the short telomeres and the absence of telomerase don't stop the tumor from forming, but rather they stop the growth of the tumor. And it's the growth of the tumor then that leads to the death of the animals. So again, it's not the loss of telomerase, but rather it's the short telomeres that block tumor growth. If it were the absence of telomerase, then you would see this effect in the G1 null animals. So it must be the short telomeres that are causing this effect. So while we were studying a number of these implications of telomerase in cancer, we also got very interested in the role of telomerase in normal tissue development. As I told you, the initial characterization of the telomerase null mice indicated that in tissues of high turnover, that there may be some defects in the mice. And while we were characterizing our telomerase null animal, this paper was published. It was a collaboration of the Dokel lab with the Bessler and Mason labs. And the paper was entitled, "The RNA component of telomerase is mutated in autosomal dominant dyskeratosis congenita." So this is a human inherited disease. I'll tell you about the actual disease features in a minute. But what this group was doing was taking, using human genetics and linkage analysis in large families that had this inherited human disease and identifying an interval that was always segregating with disease. And then they came upon the gene and found out that it was the telomerase RNA that was the causal effect of this inherited human genetic syndrome. And so we got even more interested in the role of telomerase in normal tissue based on this. So some of the clinical manifestations of dyskeratosis congenita. The disease gets its name because there are skin and nail problems. So you can see abnormal nail growth as well as hyperpigmentation and oral leukoplakia in the mouth. So these are the presentations and the reason that the disease gets its name, dyskeratosis, because what the doctor initially sees are these skin manifestations. Although it was well known at the time that the mortality of the disease was primarily due to bone marrow failure. So that's not something you can see visibly, but it is a bone marrow failure syndrome. And so this made us think about this hierarchy of the stem cells in the bone marrow, which gives rise to all of the different components of the bone marrow. And I'll tell you in a minute that it's not just bone marrow failure, but there are very important other organ failures that occur in these telomerase mutant families. So the autosomal dominant inheritance of dyskeratosis congenita was very interesting to us. So there was a physician, Rob Brodsky at Hopkins, who called me up one day and said that he had a family that had this inherited syndrome that had just been identified to be due to telomerase. and was I interested in studying this family? And since I'm not a physician, I turned to a collaborator, Mary Armanios, who took this proband, who came into the Hopkins Clinic, and got very interested in the family, and was able to bring this 3-generation family to Hopkins and do a complete physical phenotype to understand what was being inherited here. And we were very interested in this because it's inherited in autosomal dominant fashion. So what's shown here is the grandmother and grandfather of the proband, and then the father of the proband. And each one of these symbols that's shown is in black. What Mary found was there was not a mutation in the RNA component of telomerase, but rather in the protein component of telomerase, TERT. So each one of these individuals had a TERT mutation, and the affected individuals are then shown in the black. The interesting thing about the autosomal dominant inheritance is that it also shows genetic anticipation. So what genetic anticipation means is that with each generation in the family, there's an earlier onset of disease and a greater severity of disease with each generation. Typically, if you read in a human genetics textbook, that will be attributed to triplet repeat expansion. So for instance, in Huntington's disease and other inherited diseases, where you actually have an expansion of these repeat triplets. And what I'll show you is that now telomere shortening is the second molecular mechanism that can explain this genetic anticipation. So the earlier onset of disease and the worsening of the phenotype should remind you of our telomerase knockout mouse. So there's genetic anticipation, but the affected people are heterozygous for mutations in either the RNA or the protein component of telomerase. And this was of interest to us as geneticists. If you think about how that might play out, you can imagine if you have one wild type allele and one mutant allele, perhaps the mutant product would dominantly negatively take out that wild type product in a dominant negative fashion that could then cause this inheritance to show the phenotype in the heterozygous state. The alternative to a dominant negative effect is haploinsufficiency. And that is you have one wild type allele and one mutant allele, and it's just not enough to have only one wild type allele. And so what I'll show you is we were able to go back to our mice and show that indeed it's haploinsufficiency that underlies this inherited genetic syndrome. So what we were able to do now is to go to our telomerase heterozygous mice, and when you cross 2 heterozygous mice together, of course, you get out the null animals, but you also get out the heterozygotes. So we were able to designate this the first generation heterozygote mouse. And then we cross 2 HG1 mice to each other, and we generate heterozygous generation 2 mice. We cross the HG2, get HG3, HG4, etc. And what we were able to show is that there was specific telomere shortening with each generation of the breeding of these heterozygous mice. So when you have half the level of telomerase, it's not enough to be able to maintain the telomeres through many generations of breeding. So this was somewhat of a surprise, although it mimics the human disease. Some people might have thought that with each generation, when you go back and make a new zygote, that then you would fix the telomere problem and then you would be able to go from the next generation. But that's not the case. Having short telomeres, those short telomeres are passed down through each generation progressively, and the telomeres shorten with each generation. When we then look at the viability of these mice, again, a Kaplan-Meier survival curve. Here's the number of days and the percent of mice alive. And if you just focus on the wild type here, the wild type mice, the survival curve is here shown up to 400 days. And the HG10, where you can see that there's a significant decrease in survival, and there's a progressive decrease with each generation of the heterozygous mice that's very similar to the genetic anticipation that's seen in the human disease. So the inheritance of short telomeres decreases the survival, and it resembles the genetic anticipation seen in the telomere syndromes. And the primary cause of death is a combination of the GI defect with the immune defect that's present due to the bone marrow failure in these mice. And that's in the case of the mice, that's a primary cause of death. So if you look in the human population, at telomere length, we showed about 15 years ago, and it's now been shown by a number of other groups, that if you look in white blood cells in normal people with increasing age at telomere length, what you see is that there's a progressive decline in telomere length in white blood cells. But the other thing that's quite striking is that there's a very large distribution in the normal human population of telomere length, quite amount of heterogeneity. So if we want to really be able to study what happens in the human population when telomeres get to be short, one needs to have age-matched controls and be able to know where an individual is along this distribution of telomere lengths. So Peter Lansdorp in Vancouver was able to adapt his Q-fish assay that I showed you earlier and to apply it as a flow fish, where you hybridize the white blood cells and now you put them through a flow cytometer and determine what is the signal intensity of the telomere length in the normal population. And he was able to take such a distribution and looking at a large number of normal individuals, put confidence in intervals on that. And so here we have the age and the telomere length and the decrease. And so this would be the 99th percentile long in the population and the 1st percentile short. And so in order to study the families that have these telomere syndromes that are coming into the clinic at Hopkins, Mary Armanios was able to use about 200 normal individuals at Hopkins and plot that in the dotted lines in comparison to what the Lansdorp lab had shown in Vancouver. And you can see that there's very good correlation between these. So now we have an assay that's a very robust assay where when an individual comes into the clinic, we can determine what is their telomere length. And what Mary has found is that now that we have this clinical assay, there are people referred from across the United States and in fact across the world, and she now has a large registry of patients. And if you look at these telomere syndrome patients on this percentile ranking here, you see that the telomere patients are significantly shorter telomeres than the normal population. And indeed, what Mary has gone on to show is that it's not just a bone marrow failure syndrome. By taking all of these patients and looking very carefully within the families at what the diseases are that are segregating in the families, she's been able to show that, yes, there is the bone marrow failure syndrome, but the most prominent effect is actually pulmonary fibrosis. Typically, one is referred to this as idiopathic pulmonary fibrosis. Idiopathic because it wasn't known what caused it, although it's very clear that this short telomeres underlie this syndrome. We did not see this in our mice, and we can talk about this later at the end. Mice only live to be a couple of years old. Idiopathic pulmonary fibrosis comes on in the 60s or 70s in the human population. There's predisposition to emphysema as well as pulmonary fibrosis, cryptogenic liver cirrhosis, as well as GI disease, as we had seen in the mice. So now we can use this registry of families to try and understand what actually happens in the human situation when telomeres get to be very short. So Mary has gone on to show, very interestingly, that the youngest individuals that have these telomere syndromes have immunodeficiency. Bone marrow failure also occurs in the young individuals. The intermediate individuals have liver disease, and it's the older individuals that have pulmonary fibrosis. So there's a very interesting pattern of disease that occurs in this situation. I've told you that there's genetic anticipation. So genetic anticipation is a case where here you start off with long telomeres, and the next generation the telomeres are a little bit shorter, and the next generation the telomeres are shorter. And so you see a worsening of disease with each generation. But in the case of the telomeres, there's also an evolving pattern of disease. So the earlier generations have this pulmonary fibrosis, the older individuals, and when that allele is then passed on to later generations and later generations, there's a different phenotype that occurs. And so by using the genetics, you can see that it's actually the exact same allele that causes pulmonary fibrosis in the earlier generations that then causes the bone marrow failure in the later generations. It's not the case that there's locus heterogeneity and a different mutation in the gene causes a different kind of disease. It's the how short the telomeres actually are and how it manifests in disease. So this is a very interesting evolution of disease. So as I've tried to point out, it's the short telomeres that are causing the disease in these situations, both in the mice as well as in the human telomere syndrome patients. And of course, if this is the normal distribution in the population, those individuals that are in the normal distribution but at around the first percentile are at risk for a number of these diseases, even if you don't have a telomerase mutation, because it's not the mutant telomerase that causes the effect, it's the short telomeres that cause the effect. So that's the reason that a number of these diseases are very similar to age-related degenerative disease. So we're very interested in understanding how the normal telomere length distribution is regulated, because it's so important what that equilibrium telomere length maintenance is, because the short telomeres will cause disease. So what we know is that the proteins that are bound to the end of the telomere have to somehow interact with telomerase to establish this length regulation. So telomerase will lengthen the telomeres, and then DNA replication will shorten the telomeres, and you somehow magically establish this equilibrium. And how this equilibrium is established and maintained, we know very little about. We know a lot about the identity of these particular proteins, and we know about the components of telomerase, but how this is regulated, we really don't know a lot about. So that's what we are really focused on in my laboratory now, is to try and understand how is this length regulation normally regulated. So if you want to study human telomere length regulation, the standard way people have measured that in the laboratory is to do a southern blot like the one shown here. What this is human DNA restricted and probed with the telomeric probe. So you see this heterogeneity that reflects this length distribution here. And when you take these cells, if they don't have telomerase, and you passage them for the increasing numbers of population doublings, you see that there's a decrease in length and a decrease in hybridization to the probe. So this is the standard assay that's used. And so 88 population doublings is about 6 to 8 months of cell culture. We wanted to find a way where we could manipulate telomere length equilibrium in a way that wouldn't take us 8 months in order to be able to see a change. And so Stella Lee, who was a graduate student in the lab, adapted an assay which had been originally probed in yeast by Dan Gottschling's lab. And we called this the ADDIT assay, or addition of de novo initiated telomeres, as a way to measure telomere elongation. So what Stella did was to engineer a mouse chromosome where endogenously there's this hygromycin sequence, and then about 2 kilobases of telomeric sequence, which we call the seed sequence, and then an ISCE1 cut site. This is a 13 base pair recognition site for an endonuclease. So when she then expresses under inducible condition this ISCE1 endonuclease, it will cut in vivo at this site, and then telomeres can be added by telomerase, and then we can either knock down with siRNAs or inhibitors, or do things that we think might increase telomere length and measure telomere length in vivo in a way that would take about 24 hours rather than 6 months. And so when Stella set out to do this, she started out with cells that were either telomerase positive or telomerase negative, and designed a way that you can ligate an adapter onto the ends of the chromosome. This was called a teloret. We adapted this from another group who used this as a way to sequence human telomeres. So you can adapt on a linker here, and then use a probe within the hygromycin sequence and within this adapter here. And you can do this either in the presence of telomerase or the absence of telomerase. And then as a control, we've designed a primer that we could ligate directly to the cut ISCE1, so we could know how long that product is before it's been changed in vivo. So I'll show you here, if we just focus on this F2 primer shown over here, the control lane is shown here. You see this band just above 1.5 kb, which represents the length of this fragment here. Now, when we do this in a telomerase positive cell line, you see this smear that extends above the length that we initially input. And when we do this in the telomerase negative cell line, we see the smear is predominantly below the cell line. And very similar when we use a different primer. So this was very exciting that we could see in 24 hours a change in the PCR product length. However, how many of you in the room have done PCR? Okay, how many of you have ever seen a PCR artifact? Same number of people, probably. So while this was really exciting, we wanted to really be sure, is this really what we think it is in terms of the elongation of this cut site? And so what we decided to do was to use PacBio sequencing, Pacific Biosystems, where you can get very long sequence reads. Unlike traditional deep sequencing where you have very short reads that you align, the PacBio would allow us to sequence all the way across this, and we use the criteria that the products had to have both the hygromycin sequence and this telltale sequence. So we knew that they had to be the full length sequence. So when we do that, we did that either in the control situation, the telomerase positive, or the telomerase negative. So I'll show you first in the control. What we've done is taken hundreds of sequence reads and aligned them here at the junction between the hygromycin sequence and the telomeric repeats. And the hygromycin is shown in blue. All the telomeric repeats are shown in orange. The ISCE1 cut site is in green. And then all of the products have to have both hygro and this linker sequence. And so what's shown in the darker orange are slight mismatches to the telomeric repeat. PacBio sequence is well known to have about a 15% error rate of insertions and deletions. That doesn't matter to us because we don't need to know the sequence. What we want to know is to use this as a ruler to know what the length is of this product. So then we can do that in the absence of telomerase or in the presence of telomerase. And what you see here in the presence of telomerase is you see some cases where you have the ISCE1 site, and then there are telomeric sequences that are extending beyond that ISCE1 site. So we were very fortunate that the ISCE1 site itself, if you actually zoom in and look at these sequences, what's shown in red is the telomeric seed sequence that we put in, the TTAGGG. Here is the ISCE1 cut site, and after cutting, what it leaves is TAGGG, which is very much like a telomeric repeat, so telomerase can get onto that and elongate that sequence. So in these cases, what you see is telomeric repeats extending in the exact correct register of the sequence beyond that, showing that it's a templated addition of repeats onto that ISCE1 site. So what we can now do is to use the percent of reads that have the ISCE1 site and go beyond that, and we know that those must have been added by telomerase. So our criteria for de novo elongation is reads that are after the ISCE1 cut site, and we can look at the percentage of those, and it's around 20 to 25%, usually in the case of the telomerase positive. There is some slippage due to PCR as well as PacBio sequencing. So in the MTR null cells, you do see a few of these long reads, but in no cases do they have the ISCE1 cut site first and then the repeats. So that's why this is 0% here. So now we have an assay where we can actually intervene and look at telomere length, and it's about a 24-hour assay, and then the PacBio sequencing. So I'll just show you one example of how we've used this. We have a number of experiments underway to really try and understand how the telomere length equilibrium is established. But the first thing that we set out to look at is the role of ATM in telomere elongation. So ATM is known when you have a double-strand break that activates the ATM kinase. And what happens is you get a cell cycle arrest that can then allow for a period of repair, or under some circumstances you can get cell death depending on the amount of damage or the cell type. So I showed you already that if you have progressive telomere shortening, that signals a DNA damage response, and we know that that goes through ATM. And our question is, although we know this can cause senescence or cell death, Could there be also something analogous here to the repair pathway that's known in the DNA damage area? So the role of ATM at human telomeres has been really quite controversial. It's been known in yeast for a number of years that the TEL1 gene, which is the ATM homolog in yeast, does regulate telomere length. In mice, there is no evidence for ATM in telomere elongation, but these laboratory mice have extremely long telomeres. And papers looking in human cells, some have said that ATM is involved in telomere length regulation, and others have said ATM is not involved. So we thought that we would take this as a first example of asking the question, what is the role of ATM in telomere elongation? So what Stella did was to have two ways to inhibit ATM. There's this drug that we will call the COO drug, and also she used siRNA to specifically inhibit ATM. So what's shown here is cells that were either not treated or treated with DNA damage, camptothecin. And when you treat with DNA damage, ATM will phosphorylate this particular protein CAP1. So you see this very nice ATM-induced phosphorylation. In the presence of this drug, that is not phosphorylated. And in the case of the siRNAs, again, no treatment with camptothecin doesn't activate CAP1 phosphorylation. But when we treat with CAP1, camptothecin, you have the phosphorylation, and then increasing siRNAs decreases the amount of phosphorylation, showing that the ATM inhibitor is actually working. And so when we do that ADDIT assay in the presence of the ATM inhibitor, again, in the control cells, we have about 25% addition. The drug has a decreased amount of addition, and the siRNA, we saw no addition onto the ends of the telomeric repeats. We don't yet know if this is a difference between having the protein present but inhibited or having the protein be absent. We're still following up on that. But clearly there's a role for the ATM kinase in the elongation of telomeres by telomerase. But of course this was a new assay that we had just developed. So it was important for us to show that this wasn't something that was just an artifact of our new way of doing it. So then we set out to test the role of ATM using more conventional assays. And so what Stella was able to do is to generate a virus that expresses both components of telomerase, the lentivirus. When she infects cells, we can then measure telomeres at day 0, day 2, and day 5. And by having this overexpression, it turns out that we can accelerate telomere elongation by overexpression. So again, here's a conventional southern blot showing the control DNA, and then the DNA that's treated with the lentivirus and DMSO. And you can see that there's this elongation of the telomeres over the short period of time. But when we add the COO drug, it blocks that elongation, demonstrating by a second method that ATM is essential for telomere elongation. This was all in mouse cells because her cell line that she's been using was derived from mice. So when we went on then to ask the question about human cells, and we went back now to our long 6-month cell culture assay to verify our results. And we grew cells for increasing number of population doublings, up to 75 population doublings, in the presence of a low level of this COO inhibitor. And you can see that in these human cells, there was also a decreased telomere elongation in these, or decreased telomere length in these cells. So our conclusion then is that very much like what occurs in human cells, where you have either cell cycle arrest, senescence, or that arrest leading to repair, that in the case of the telomeres, there's cell cycle arrest, but then there can be also a signaling of repair to increase the telomere length. And what it is that is exactly being regulated here, how ATM specifically allows the elongation of the telomeres, is something of great interest in the lab. So to summarize, what I've told you is that telomere length equilibrium has to balance enough cell divisions with too many. I've told you a story about telomeres. When they get too short, you can have stem cell failure and these syndromes of telomere shortening in humans. And in the case when telomeres are maintained continuously, there is an increased risk of cancer in terms of the cells being able to grow indefinitely. And so this normal telomere length equilibrium then has to balance these two sides of the roles of telomeres. And then finally, new discoveries come from unlikely places. We didn't set out to understand the role of telomeres in cancer or the role of telomeres in pulmonary fibrosis. Rather, we were just curious about how telomeres were maintained. And we went to the source, tetrahymena, where there's a whole bunch of telomeres, and that's where we initially identified telomerase. And then by continuing to follow our curiosity, we then came upon the role of telomerase in cancer and also in degenerative disease. And so the take-home message is curiosity-driven research provides unexpected discoveries that can have important implications for human health. So my final summary, short telomeres signal cell death or cell senescence after many divisions. Short telomeres limit the growth of cancer cells. Short telomeres cause a spectrum of degenerative diseases that are collectively called telomere syndromes. The ADDIT assay provides a powerful new tool to rapidly assess telomere elongation, and ATM is required for telomere elongation. And I just want to thank all of the members of my laboratory. These are the current members of the lab and my collaborator, Mary Armanios, and her group, where they have worked out many of these aspects of the human telomere syndromes. So thank you.
So with that, either who would like to start?
So I have a question. You said that it's interesting that in the early G1 and G2 divisions, you don't see any major global defects. But did you look at specific cell lineages, like the ones that divide a lot during the lifetime, like lymphoid lineages, epithelia, gut, and so forth? Because they would have gone through hundreds of divisions.
Yes, we did this all blinded with people in the pathology department, and we gave them blinded mice of all of the generations, and they saw nothing at all remarkable in any of the blood cell compartments or any of the other compartments, the seminiferous tubules. Those phenotypes were all only when the telomeres got to be very short. They could find no pathology in blinded studies.
Can I ask one more? So in your final assay, when you cut with IC1, right? You would expect that basically mimics a double strand break response, and you would bring in ATM and everything else that goes with it. But do you know if that would actually happen in a normal telomere? So do you see ATM bound there? Do you see DDR response factors in normal physiological conditions?
We haven't done any chip analysis of those ISCE1 cut sites, but of course there are telomeric repeats along the length, and what the telomere binding proteins typically do is to distinguish a break from a telomere. So one thought would be that if you have all those telomere binding proteins along there, that could help distinguish it. We actually did spend a lot of time asking in tissue culture, could we see, for instance, a p53BP1, just one site? But actually, background levels of p53BP1 in most tissue culture cells are 6 or 7 spots. That's what its background is. So we weren't able to specifically see a DNA damage, and we haven't done any chip analysis. It would be an interesting question.
I have a question to the math. So the end replication problem kind of poses that polymerase can't replicate the ends. The other behavior number tells us that somehow the cells don't divide more than 40 to 60 times. Even stem cells divide in generations in the mice in the lab at long telomeres. So does it really match that you can find out the 6 generations shortening which cause all these problems?
Yes, I mean you can do the math in a number of ways, but certainly in cell culture, if you have cells that are undergoing cellular senescence due to the Hayflick limit, and you add telomerase and you elongate telomeres, you rescue those cells. So that shows experimentally that the telomeres play a role in the cellular senescence. I can certainly tell you that in terms of the mice, standard laboratory mice, I took those slides out here, have this about 100 kb telomeres. That's C57 black 6, FEBN, all the standard laboratory models. When we wanted to ask the questions about haploinsufficiency, we used a mouse called Mus castaneus, which actually has wild type mouse telomeres. If you catch mice in the wild, they have about the same telomere length as humans, which is about 10 kb. And Castaneous telomeres are 10 kb. And when we do the knockout in the Castaneous, we see phenotypes in the second and third generations. So it does correlate with the length of the telomeres in the different mouse models.
I have a question regarding the haploinsufficiency. Is this is this related to the fact that telomeres is expressed at relatively low levels in general, or does this tell us something that the telomere, so not only you cannot replicate the end and you need to replace that, but this is maybe being degraded very rapidly and you need to counteract that, or both? Or?
We know that telomerase is expressed at extremely low levels. The estimates in human HeLa cells, so cancer cell lines, are about 100 copies per cell, even in a cancer cell line. And so in normal human tissues, it's also very, very low. In yeast, there's about 12 copies per cell. So all of these organisms, telomerase is really limited. And that may have to do with this balance, is that there's just enough telomerase to be able to do what you need to do. And then when you have less of it, there becomes a problem. But it's really quite remarkable in these families. You can clone out the mutant telomerase, and sometimes that telomerase enzyme will have 70% enzyme activity if you measure it in vitro. So that means those people have one wild type allele and one allele that's 70% active. And in many generations in the family, that then still gets to have short telomeres. But of course, you would expect it to be many more generations than a null allele in the telomerase.
So is the sequence of the telomerase repeats is very conserved?
Yes.
But is that mainly for the telomerase RNA to be able to prime, or does that conservation conserve other sequences? So if you introduce the tetrahymena repeat in the telomerase RNA in mouse, does that then?
It causes an immediate DNA damage response, and that is because those proteins that are bound along the length are sequence-specific DNA binding proteins. So in humans, that's TTA, GGG, and if you change it to the tetrahymena sequence, then now those proteins don't bind, and you've suddenly deprotected. So you have your normal telomeric sequence, and now you add your mutant on here, you've now suddenly deprotected the end. And so those cells die. It causes rapid cell death.
Is there a correlation between the age of the father and the anticipation effect?
There have been a number of studies in humans which have looked at age of fathers and the telomere length in the offspring. And there seems to be a pretty good correlation across a number of different studies that indicate that the children of older fathers actually have somewhat longer telomeres. And the thought is that perhaps during the reproductive cycle over a number of years, in the testes there actually is some telomere elongation. There is some evidence for this. But of course, we also know that older fathers accumulate DNA mutations. And so, although it was published in Science magazine, wouldn't it be great if we could just have older fathers and then you would have longer telomeres and you would have less disease? Actually, I scratched my head when I read that because it's well known that because of the errors in DNA replication, that there's increased genetic disease in older fathers. So I think that that's probably not a way that we want to go.
So over the lifetime, you showed that over a lifetime of humans, basically there is progressive shortening.
In white blood cells.
But does that mean the enzyme telomerase actually wears out, or how do you explain that?
So we know that at first it was a mystery why there is this telomere shortening in white blood cells. And before we had a way to assay telomerase, we thought, well, maybe they don't express telomerase. But you can look and you can specifically purify CD34 positive stem cells, and they do have telomerase activity. But the thought is that because telomerase is limiting, you have just enough telomerase usually to do what you need to do for most of the cells in the body. The number of cell divisions that occur in the hematopoietic system, you slightly outstrip the amount of ability of telomerase to maintain it. For most part, it's a buffer zone. For most of that heterogeneity, it doesn't matter. It's only when you get down to really the least percentile that you have a problem. And so that's what we think is occurring in the shortening in the white blood cells. If you look, for instance, done in mice at neuronal tissue with age or various things like that, you don't see that change because they're post-mitotic. So post-mitotic cells don't show that.
Last question from Andy.
So it seems to be most important for an organism to maintain telomere lengths in the germline. So I was actually wondering whether there are specific mechanisms then in the germline that would actually maintain the telomeres at a certain length, because also in your kind of course of generations you see like this progressive shortening, but that doesn't seem to be due to the somatic cells that actually happen to the shortening, but it must be.
Certainly, people have thought that maybe in the germline, or when you have a zygote formed, that now in the early embryo that you may have a lot of telomeres that will then fill out the telomere problem. But genetically, that's clearly not what's happening, because in the mice that are heterozygous, you have just half the level of telomeres. The short telomeres go from one generation to the next, and they go through the germline in each one of those generations. And in the human patients, there's progressive telomere shortening in those families. So there isn't any resetting in either the germline or the embryo, or you wouldn't see that progressive telomere shortening. So that's what the genetics tells us.
Great. Well, thank you very much again, Carol.